You're not like you,
I would like to extend a warm welcome to Professor Sharat Chandra to CCE today for Viva. Professor Sharat Chandra is very popularized research in molecular genetics, in particular human genetics in the country, well, way back from mid-70s. And particularly in the area of human genetics, he not only stressed but sensitized people, showing the importance of genetic analysis addressing diseases in humans, and particularly genetic links, and was really one of the first to do genetic counseling in the country as way back as in the mid-70s. And this is something which is very dear to him, and he's also popularized this in the country to a very great extent. Professor, we are very fortunate to have Professor Sharat Chandra as Director CCNB. And so he is really from three places, or two places I would say, from the Department of Microbiology and Cell Biology, Indian Institute of Science, and from CCNB. He is currently in the Center for Human Genetics. So, this candidate, Parmit Kumar Singh, did his master's in biotechnology from the University of Pune, and joined Kazbekar in early 2005 for his Ph.D. work. His thesis has been examined by Professor Sharat Chandra and Professor Bharat Chattu from the Department of Microbiology and Biotechnology Center, MS University, Baroda. Both have commended the thesis. And Professor Bharat Chattu has remarked that the work of the candidate has made further contributions to our knowledge in this area, and the work has been presented in a very systematic manner through many elegant experiments. And we would like to compliment the candidate for a very nice piece of work of very high quality. Professor Sharat Chandra also remarked that the thesis illustrates the intricacy of the field and how meaningful advances are being made by careful experimentation. The experimental results presented in the thesis are clearly described. The illustrations are excellent, the presentation is good, and it has been a pleasure to read this thesis. So Parmit would current research topic is also in the area of mapping of suppressor of meiotic silencing in a wild isolated strain of Neurospora. So he continues his interest in this area, and of course you all know how we go by. So we have a short presentation for about 30 minutes by Parmit, followed by a question. So everyone knows the format. So
thanks for the kind introduction. Good afternoon, everybody. So the title of my thesis is Genetic and Molecular Analysis of Neurospora Duplication and Duplication Genetic Translocation. So Neurospora is a fungus, and it has two matting types, mat DJ and mat radius. So when ascospores germinate and produce hypha that differentiates and produce conidia that acts as male gamete, and under nitrogen limiting condition it differentiates and produces female structure called protoperficium. So fertilization of protoperficium by conidia of opposite matting type triggers its further differentiation into perficium, where the two nuclei of opposite matting type continue dividing and before carried for short time, 10 to 15 times, and then the after carried the zygote undergoes immediately meiosis and mitosis and forms 8 spores, which is enclosed in eschers. So these are in two mating types. Spores are bigger and spores are littler, and these ascospores are ejected forcibly from the beak of the patricium, and then again it germinates under favorable conditions and the life cycle continues. So in the lab we grow Neospora in a small petri dishes, and the vegetative structure looks like this, and the produced quantity are covered within 2 days the full area, and this is hypha, the growth of, and then the reproductive system looks very black under microscope, and this small is picking up on that. So coming to duplication and translocation. So in Neurospora, crosses heterozygous for translocation produce duplication progeny. So this is the picture of translocation. So in translocation, a segment of second chromosome is inserted in first chromosome, and when this strain participates in a cross with normal strain, then due to alternate segregation, it produced parental combination, and adjacent one segregation produced duplication and deficiency. So deficiency are inviable. So 1/3 of viable spores are duplicated for this translocated segment. So in a duplication heterozygous cross, during meiosis, this translocated segment will be unpaired. So in Neurospora, it is known that unpairing of DNA triggers meiotic silencing, that is RNA-based pathway, and meiotic silencing silence all the genes that fail to pair during meiosis. So, and your homolog copy which are paired on paired somewhere. So SAD1 and SAD2 are two genes known involved in meiotic silencing, and SAD1 encodes RNA-dependent RNA polymerase, which makes double-stranded with aberrant RNA. So the aberrant RNA are produced from the unpaired DNA, and it's transported into cytoplasm where it makes double-stranded RNA and choked by dicer, and further the silencing occurs. So SAD2 encodes a protein which recruits SAD1 in the perinuclear region, which is essential for SAD1 function. And so, for example, when a cross in this cross, where the wild type is crossed with a strain containing transgene GFP, there is no expression of GFP in the site during meiosis because the GFP fails to pair, and that unpairing of GFP triggers meiotic silencing and it silences GFP. So semi-dominant mutation of SAD1 and SAD2 suppress meiotic silencing, and then there is GFP expression in the site. So these are 8 spores during early meiosis, and they are shown. So if the genes are essential present on this unpaired DNA, are essential for meiosis and downstream process like ascospores pigmentation and ascospores maturation, then the crosses are barren in Neurospora. So barren, so we identified in laboratory the duplication heterozygous crosses by barren phenotypes where the cross forms normal perithecia, but they produce only 100 and 200 ascospores, whereas the fertile cross produce 1,000 and 10,000 spores. So the translocated DNA of the duplication will be unpaired, and that triggers meiotic silencing, and it will be silenced. So again, the suppressor of meiotic silencing suppress duplication, meiotic silencing, and then restores the fertility of duplication heterozygous process. So these alleles, the semi-dominant allele of SAD1 and SAD2 are division alleles, and they cause unpairing at that locus of the wild homolog copy, and thus they self-silence actually, and that's why the meiotic silencing didn't operate for these translocated segments. So duplicated DNA in Neurosporas are substrates for repeat-induced point mutation, which is another genome defense process occurs during dikaryon stage before carriogamy in the sexual stage, and REAP mutates any DNA duplicated DNA which is greater than 400 base pair in size. So when duplication participates in a cross, the REAP machinery detects duplicated DNA during the dikaryon stage and mutates the sequences by converting GC into AT, and the unconverted cytosines are methylated. So it converts GC into AT and the cytosine methylation. So The other nucleus where there is no, the sequence is not duplicated, so that will be unaffected. So it affects only the nucleus where the duplicated DNA is present. So as I told that this read mutates both large and small duplication. So we use these two mutated genes, and this strain called as EPR3 has a copy of 1.3 kb sequence of I just told 3 gene, which is present on linkage group 3 in wild type. So the crosses which are heterozygous for DPR3 produce 20 to 30% ERK3 in a F1 generation, and escospores mutant in ERK3 gene produce different produce colonies which have different morphology compared to wild type on the servos containing media. So under microscope we calculate this colony, which is ERK3 mutant colony. They are very compact and slow growing, and calculate the ERK3 frequency and use this as the read passage. So as I told that both the large and the small duplications are substrates for read. The crosses which are heterozygous for large duplication and DPR3, it was found that such cross produced less than 1% ERK3 frequency when the size of large duplication is greater than 220 kbp. But when the size of large duplication like DPP362 is just 119 kbp, so crosses heterozygous for DPP362 and DPR3 produce greater than 1% ERK3 frequency. So only 6 large duplication were used, and one of them B362 was non-suppressor. So it suggests that large duplications are able to suppress repeating a small DPR3 duplication by titrating the repeat machinery. And so there should be a threshold limit between this 119 kb and 270 kbp and for the suppressor. So I increased this number to 37, and I found that out of 37, these 34 duplications were suppressor, and I estimated the size of these duplications. I found that all these duplications were greater than 270 kbp, and I found these 3 smaller duplications, smaller than 200 kbp, were non-suppressors. As a control, I did all the parental translocation crossed with DPR3, and the F1 non-duplicated progeny also I crossed with DPR3 and estimated the ERK3 frequency. And in this case, the ERK3 frequency was greater than 1%. So it's clearly the size of large duplications are affecting the repeat, the small DPR3. So finding of more than one non-suppressor here and estimation of their size, that they are smaller than 200 kbp, allowed me to do the crosses which were multiple heterozygous for multiple non-suppressors. And I found that when the combined size of these small duplications approach towards 300 kbp, then the crosses started showing suppression. So like here in this cross, which was heterozygous for DPV4 and R23 and 4, the combined size was 305 kbp. 1 out of 11 crosses, I found less than 1% ERK3 frequency. And when the size was 3 to 0 kbp in a cross which was homozygous for DPR23 and 4, 3 out of 6 crosses showed less than 1% ERK3 frequency, and it suggests that threshold size of the suppression is near to 300 kbp. So I determined the size of large and small duplication by PCR-based RSLP approach, where I crossed degenerating translocation, parental translocation with 10 different wild isolated strains collected from different geographical parts. So these wild isolated strains I used because they have RFLP as compared to this translocation, and all these translocations were generated in wild type background. So the duplication progeny were identified on the basis of parent phenotype, and any marker which is present on translocated segment, like here 8 marker. So for that marker, duplication progeny shows both the presence of both the alleles, so AN and AT. So this is known as covered. For any marker which is outside of translocated segment, the duplication progeny shows one